Welcome again everyone to G Squared Academy, where excellence is as usually epitomized. As you can see on the screen there, and I'm not going to delay too long, because I know that people are pressed for time, really. But first I want to thank everyone who has subscribed to the channel, who has watched the videos, anyone who has interacted with the videos, so on and so forth, shared, Thank you very much for the support. Alright, just keep supporting. And I realize that most of those you viewers, you watch the videos but you're not subscribing, so just hit that subscription button for me now, please, and touch the post notification bell so anytime I post a video you can get it. Alright, so as you can see on the screen, so without further ado, I'm going to be looking at the CSEC biology paper 3, and it was actually requested by somebody who watched the video. I just want to do this video so you can help prepare for your exam. Okay, so first of all, the paper 3, which is what you're going to be doing, it replaces the SBA. So some students, for whatever reason, just cannot do an SBA. Whatever their situation is, we don't know. They just can't do an SBA. So you have to remember that the paper 3 now replaces that SBA, which means that you have to cover material which other students who get to do their SBAs, what they did in 2 years, they have to do it in like one paper, really. So you still have the skills. It's a practical paper, really. You have no SBA, so you still have to work with the skills for biology, ORR, observation, recording, and reporting, manipulation and measurement, drawing, analysis and interpretation, and possibly planning and designing. Okay, since it's a practical paper, your examiner really tests your manipulation and measurement skills, your drawing skills, your ORR skills, your AI skills. P&D is a little bit more ticklish, but not that it is impossible. But it's those 4 ones mainly that you would be tested on. Alright, so for this year's paper, I'm looking at a certain number of topics: food tests, ecological sampling techniques, the eye to some extent, and disease. To be honest, I'm kind of flabbergasted by the fact that the eye and diseases are in a paper 3, because I'm thinking, what practical would you use for those things? And I mean, maybe the pinhole camera. Making the pinhole camera is one of those things, right? Or you could draw the eye as well, for as a drawing skill, or making a pinhole camera. But apart from that, really, it is weird that those 2 topics, diseases and eye, are on this paper. But nonetheless, let's see what they come up with. Alright, so for your food test, I would say it's best if you use a table. So let's say they give you a question about food tests and what you should be doing and how you test your food samples. I suppose it's best if you use a table. So what would you really do, right? You get your nutrient that you're going to be testing, the method of carrying out the test, and then your positive result. How do you know if this nutrient is present? So let's take an example. For starch, for example. Alright, so starch, you're testing for starch. What do? You're going to make a solution of your sample, if you can make a solution, or you can make a suspension of the sample, or you can just add the iodine to the sample. You could just add it to the cracker. Now let's say starch is present. What would you observe? Blue-black color. Alright, so it's pretty straightforward there. Next nutrient now is protein. So you want to test for protein. What is the procedure? What do you do to test for protein? So the first thing you need to do is you need to add 1 gram of potassium hydroxide. It could be sodium hydroxide as well, because potassium and sodium hydroxide is pretty much the same thing. And then when you add that to the sample now, you are going to stir and then add 1 drop of copper sulfate solution. Copper sulfate is blue. Now, if protein is present, what you see is purple. In some textbooks I saw mauve, but seriously I really don't know what that is. But I know what purple is. Okay? So that's what you would see. Now for fats, let's say they say you need to test a sample for fats. What do? You have a number of options here, right? You could do the spot test. So for the spot test, what you're going to do is you're going to take the sample, and you're going to add it to a piece of paper. Usually it's a liquid sample that you have if you're testing it for fat. Right, so you take the sample, you add it to a piece of paper, okay? And then now, if fat is present, you're going to see a permanent translucent spot. Now, the second thing you could do is the ethanol test, where you're going to add ethanol to the sample and you're going to shake it vigorously. Now, if fat is present again, you're going to see a white precipitate. So you could do the spot test, where you add the sample to the paper and you see a permanent translucent spot, or you could add ethanol and shake vigorously and then you see a white precipitate. White precipitate just really means a white solid. It's nothing grander. In chemistry especially, we use the term precipitate to say that this thing is a solid. Suffice to say, just for that. Yes. Then now you're reducing sugar. You're reducing sugar test really. Now you can use two reagents. You can use Felins and you can use Benedict's, but in our region we tend to use Benedict's solution. So you're going to add Benedict's to the sample and you're going to boil it. Alright, you add your Benedict's and you boil it. Now if the reducing sugar is present, you're going to get a brick red precipitate. Alright, hope you guys following so far. So of course, as I said, it's best for you to use a table, because a table organizes your information and your data better than just writing them in paragraphs or writing one test, then the method, then the results, as you can see here. Okay, so your nutrients, so what you need to know doing your paper 3 is, what nutrients should I be testing for? These are the nutrients that we usually test for: starch, protein, fat, and reducing sugar. Now, clearly, if you test for reducing sugar and you don't get a positive test, it means that that substance is a non-reducing sugar. You follow what I'm saying? So in doing a test for reducing sugar is also doing a test for non-reducing sugar. All right. So these are the nutrients that we test for. This is how we test for them, and this is a positive result if the nutrient is present. All right. Hopefully that helps you. Now moving right along very briskly. Okay, the next thing now is our ecological sampling techniques. So ecological sampling techniques is a part of the exam. You're in the exam, you're given a question, you need to know which ecological sampling technique I would need to use for this question, for this particular situation. Okay, and that's what I want to show you now, how you determine these things. Okay, so here we go. The first one is capture and recapture. Capture and recapture. When would you use capture and recapture? Well, you use it for bigger organisms, and it's like tagging. So you have a big organism, usually big organism. Of course, there are exceptions. And you want to let it back into the wild, of course. You're doing ecological studies, you know. So it's not just about numbering the population. It's also about seeing how they interact with other organisms. It's seeing how their population changes. It's a whole host of things. All right. So you use capture and recapture for usually bigger organisms. And it's like tagging. So you tag the organism and you release it back into the environment. There you go. There's an organism. Around its neck, you can see the tag. Okay, so they would be able to track this organism to see where it goes over a period of time, so they could study migration patterns, for example. Okay, so that's one of the things. When you use capture recapture, you're using it for bigger organisms. You're studying the movement of organisms. You're studying the interaction of these organisms in different areas. Where do they go? Really, pretty much that's what you're looking at for capture recapture. Okay, because you're able to track them down and find them, so therefore you can recapture them. All right, next thing now on the list is quadrats. Quadrats. All right, these are used to quantify small organisms in an area. So it's not like let's say you have a football field and you want to quantify the organisms like ants, for example, on the field. It would be ill-advised that you try to go through and count all the ants in the area. So what you do is you use a quadrat, which is something like that, right? You place it on the ground, you count the number of ants or plant species or small organisms in that given area, and then because you know the size of the quadrat and you know the size of the area that you're working with, you could therefore scale or work out how many organisms are in or on the football field, for example, once you know how much is in a small area of the field. Okay, so let's say you have 10 ants in 1 square meter of the field, but the field is 100 square meter. You just need to multiply the 10 by 100 and you know how many ants are on the field, so to speak. That's simple math stuff. All right, so you use quadrats to quantify small organisms in an area, and that's an example of a quadrat right there. Okay, so our next thing, moving along, is a Puta. I know a lot of people don't know what this is, or they've probably never heard of it. Okay, but it's used to capture small organisms. And of course, remember that ecological studies are not just about quantifying populations. It's also about studying organisms as well. All right, so here's an example of a fellow using a Puta. Okay, really, so he's now capturing over here a small insect of the tree, and he's basically sucking through, creating a vacuum and pulling the insect off into the container here, where he can go and then study it. Okay, and of course this method is kind of non-destructive, because you collect the insect in the container and then you can study it and release it back. So it can be used to capture organisms and release them back into the environment. All right, so moving along. Next thing is our nets. You know, I remember in university when I saw the students walking around with these things, and it was a bit funny, to be honest. So it's used to capture mobile organisms, not huge organisms now, necessarily, but kind of relatively small organisms like butterflies and moths and those things, right? So you're trying to capture mobile small organisms, and here's an example. Of course, you couldn't use this to capture any big organism, right? You'd have to use something like this to capture butterflies and moths and maybe bats, bees possibly, and those sort of things. Okay, so mobile, that is the key thing here now. The net is used to capture mobile organisms. All right, so these fellows now, they could go to this bush area here, and they shake it, and then anything which comes out, they try to capture it in the net. Anything that flies out, they could try to capture it in the net. Okay, so key point here, mobile. Moving on nicely. All right, then you have your pitfall traps used to collect small insects, amphibians, and reptiles. Here's an example of that. So basically the organism climbs on the thing and falls into this trap. It's like a typical trap used for hunting organisms, and of course you're using this for small organisms. Okay, I suspect you may be able to scale it and use it for huge organisms like maybe an elephant possibly, or a lion or something of that nature, but usually for small organisms. Okay, I suspect also that for larger organisms like that, you'd use a tag for them, not necessarily to capture them, because you might cause damage. They may break a leg, and then that's not going to help them in the environment or in their habitat. All right, so next thing now, we're moving on, is your line transect. Okay, so this one now is a bit interesting. What you use basically, you use it when the habitat is changing. So you're moving from one habitat to another. Example, you're moving from a grassland to a forest, or you're moving from the land to the sea, or you're moving from grassland to swamp. You follow what I'm saying? So you're moving from one habitat to another, and this is where you use something of this nature, okay, because it's good to work there. And all you do, of course, here is that you have your line and you take samples of the organisms along the line. Okay, so as you can see in this picture here, they're taking samples of these flowers as they move along the line, and then you have grass, then you have flower, then you have rocks, and then you have the grass again. So you're able to see how the population of different organisms changes as you move along the line. Okay, so that's the example of. Now here's a question dealing with ecological studies. It says here, students of an ecology class conduct a field study in an area located on the boundary between a savanna and a forest. In addition to using various sampling techniques to estimate the species abundance, they observe the feeding patterns of the organisms. Describe 2 sampling methods which the students could use to estimate the abundance of plant species in the area, and that's 4 marks. 4 marks tells you that you need to name the species, 1 mark each, and then the description, 1 mark each, gives you the 4 mark. So looking at this now, you can say, okay, think about it. What have I just learned would suit this? Now look at the key thing. It says here that the area is located on a boundary between a savanna and a forest. So therefore, you have a habitat here that's changing. And if you go back just a little bit, there you go. Line transects. Use when the habitat changes from one into another. Okay, so for this question clearly, for this question clearly, you're going to be using a line transect and you describe how it is used. Okay, another thing that you could use is a quadrat because you have a savanna. Okay, so you could use it on the savanna moving into the forest area. So you could use the quadrat as well in sampling in this area. Okay, now finally, you describe one method they could use to collect data of mobile animals. See it right there, mobile animals. Now you don't have to look far, you just need to go back a bit. Here you go, mobile animals. Use to capture mobile animals. So you could say a net could be used to do that. So that's just an example of a question that you may be given and how you should approach it. So I hope this helps you guys. I hope this helps you guys in your exam, which is imminent, to be honest. I hope it will be of great use to you, and I wish you all the best in your exam. Please share the video, like it, subscribe to my channel, G Squared Academy, where excellence is epitomized, and I will see you next time. All the best.